what I'm going to be talking about today in more detail. We'll talk about the arrangements here, the graphical representations, and how to find this level of detail that I'm illustrating. So for some of you, if you've used the UCSC Genome Browser before, parts of it will be review, but typically people find that we can show them some tricks and some other details that they didn't realize, even in this introductory material. First of all, I want to give full and total credit to the team that develops this. The team from the University of California at Santa Cruz, led by David Hausler and Jim Kent, works very hard on updating this, providing new software all the time, bringing in new data all the time. And you can learn more about the different roles of the staff if you want to take a look at that on the UCSC Genome Browser site. In addition, of course, funding agencies all over the world are providing support for this because data is generated in many different locations. The UCSC Genome Browser team has solidly funded those. You can see here, you can learn more about their funding, of course, but a lot of the data is provided from researchers all around the world with various support mechanisms. Everyone can use the UCSC Genome Browser on the web for free. There are no barriers to that at all. If you are in a commercial setting, though, and you wanted to have an installation of the UCSC Genome Browser behind your firewall, and many companies do this, then you would have to obtain a license. That's a situation that would require a license. But for most people, the web browsing is perfectly fine and doesn't require a license at all. We don't handle any of the information about licenses. You would work with the UCSC Genome Browser team directly for that if that's a situation that you have. What we're going to do now is we're going to get right into the browser. We're going to talk about how to perform basic searches and find yourself in genomic regions and understand more about those genomic regions. So the first thing I would like you to see is the home page for the UCSC Genome Browser. If you wanted to follow along on a browser at this time, you would point your browser to genome.ucsc.edu. And this homepage picture is a little bit old, but I love this one because it had Darwin on it, so I've been saving this one. And it also illustrates a number of the key features that I like to point out about the UCSC Genome Browser at this point. At the top, there's general information about the projects and links to other tools that are available at the site. The lower part of the page has specific information about new features, current status of a genome, or new tools that they may have added, for example. In this case, there was a new genome that had just been released. There was a new assembly that had been released for a particular genome. There was a new tool. The in silico PCR tool was new at that time. So they'll tell you that things may have changed since your last visit, and it's a good place to look over that information and see if there's anything changed that you might be interested in. But the areas that we'll be focusing on today are the navigation bars at the top and at the side that will give you access to the features that I'm describing here. We won't have time to go through all of these, of course. There's more information available on our website about some of these other tools as well. But these are the places where you would get started with the UCSC Genome Browser. And one thing I wanted to point out is that we do have additional trainings on our website as well. We can't cover in great detail all of the things that you need to know about the UCSC Genome Browser. And there was a question from an attendee on the registration form about how to be introduced to the custom tracks and building your own custom tracks that you can use to annotate regions of the genome. And we have a specific tutorial that will cover that information. So we won't be going into that in detail today, but that's available on our site. So I wanted to point that out. We also have another training that covers some of the additional tools that we won't have time to talk about today. But what we're going to do right now to get started with the basic search is access the Genome Browser Gateway. The link at the top that says Genome, or the link that says Genome Browser at the side, will offer you the same entry point to the gateway, which I'll be showing on the next slide. So here you have the Homo sapiens Genome Browser Gateway. And at the top here is the region that we'll be focusing on. I'll talk specifically about each of these items as we go forward in a minute, but I just wanted you to focus on this point to perform the basic searches that we're talking about right now. But let me just give you a couple of other highlights of this page. This is the gateway where you would search for things like gene names, gene symbols, many types of IDs are searchable, OMIM IDs, SNP IDs, things like that. You could search by chromosome number. For example, if you type CHR7 into the search term box, you can look at all of chromosome 7 in your browser. Or there might be times where if you're reading a paper and somebody says that they found this copy number variation between these coordinates on chromosome 11, you can type in those specific coordinates, and assuming you're choosing the right assembly, you can see the same exact thing that those researchers were referring to. Of course, you can use this as a basic type of keyword search too. You could search for keywords like kinase or receptor or whatever your research focus includes.